it then results in the addition of a phosphate group to the molecule. So our 3-carbon sugar, which originally only had 1 phosphate, now has 2 on it. Questions, concerns so far? Okay, on glyceraldehyde 3-phosphate, okay, you've actually got a hydrogen attached to this carbon here. But that actually is removed. And then technically, I believe what also probably happened was they most likely brought in a water molecule and moved the hydrogens from that water molecule and added the oxygen on along with that phosphate. Well, this oxygen probably actually part of the phosphate. So we add the oxygen and the phosphate. This phosphate probably had a hydroxyl group, so one of the hydrogens comes from phosphate, the other hydrogen comes from the glyceraldehyde 3-phosphate. Okay? So this hydrogen is gone, and there was one other hydrogen as well that was part of the phosphate that's not gone. Okay. So in removing those electrons, as Trey pointed out, we're going to take NAD+. We're going to add electrons to it, reducing it, and forming NADH. Now, each NAD+ molecule can hold up to 2 electrons, and accept up to 2 electrons. So, these 2 NADH molecules accept a total of 4 electrons, but only one of the hydrogens is going to associate with each NAD+ molecule. So we form 2 NADHs, and we now have 2 free protons. Should I just start speaking in a foreign language at this point in time? Okay. Basically, what has happened so far? Okay. All that's happened is we removed some hydrogens and electrons from our 3-carbon sugar and added in an extra phosphate. Okay, so we did the old switcheroo, the bait and switch. Okay. Added on that phosphate to the molecule, remove the hydrogens and the electrons. The electrons are accepted by NAD+, reducing it to NADH. But not all the hydrogens associate with NAD+, so we're left with some free-floating hydrogens that no longer have electrons associated with them. We call them protons. Questions, concerns? Yes, Kate?
So that little swooping arrow means
That little swooping arrow means that a reaction occurred. Anytime we see arrows, it's reactions occurring. So what that means is a redox reaction occurred wherein the electrons were transferred to the NAD+ and reduced it to NADH.
You said each NADH had 2 electrons and carrying 4 electrons. Oh, correct?
Yes, that's what I said, Nurses.
Doesn't make sense.
Why not?
I don't know.
Why doesn't it make sense?
Did each hold 2 electrons and carry 4 electrons?
There are 2 NAD+ molecules, each can hold 2. So they can hold up to a total of 4 between the 2. Yes, Allison?
So this allows the enzyme thing that recognizes the 3-carbon sugar.
Sure. It's called triose. Triose. Or were you wanting me to spell it into my hands first and then say it?
Yeah.
Okay. Yeah, no, that's not going to happen. Phosphate. Dehydrogenase. Triose, Triose, phosphate, Phosphate, dehydrogenase, Dehydrogenase. I caught myself. Now, breaking it down, it's an enzyme that removes hydrogens. We've seen dehydrogenase before. Okay, whether you realize it or not, or at least have brought it up once or twice before. Okay, it's going to result in the addition of a phosphate, and it can potentially remove the hydrogen from the phosphate. And triose, it recognizes a 3-carbon sugar. This is why I've been spending so much time over the last several weeks talking to you about how important it is for you to be comfortable with breaking down terms in biology. Because if I gave you this on an exam, whether you've listened to anything I've said today or not, if you could break that word down, you can tell me pretty much what that enzyme does. Yes, sir?
Breaking down usually go from and whatever.
A lot of Latin and Greek seems to be backwards, where you can actually work backwards to get the meaning out of it. Okay? At least that's what I found, especially when it comes to biological terms. Okay, so, barreling right ahead. We've made our NADHs. Now it's time to start making our ATPs. Okay, so we've got 1,3-biphosphoglycerate, again called 1,3-biphosphoglycerate because it's a glycerate molecule with phosphate groups on the 1 and 3 carbon. That's going to be recognized by phosphoglycerokinase. Again, it's a kinase, which means it's going to transfer a phosphate, and it recognizes a phosphorylated glycerate molecule. Remember, kinase doesn't mean that it has to phosphorylate the molecule we're currently working with. It could just as easily dephosphorylate that molecule. And that's what this kinase does. It removes one of the two phosphates so that we can make ATP. Okay, so we now have produced 2 adenosine triphosphate molecules, leaving us now with what's known as 3-phosphoglycerate. Again, similar to 1,3-biphosphoglycerate, we now only have 1 phosphate, so it's phosphoglycerate, and that phosphate happens to be on the 3 carbon, so it's 3-phosphoglycerate. Maybe if I have time, I'll make a little matching exercise for y'all to do between today and Thursday, and I can have molecules named on one side and enzymes on the other, and y'all can tell me potentially which enzyme would be involved interacting with which molecule.
Don't put down.
Thanks, nerds. All right, so now we're going to take this 3-phosphoglycerate and we're going to mutate it. That is, alter the structure. It's still going to be glycerate. It's still going to be a phosphoglycerate, but we're going to move that phosphate. So we're going to mutate this molecule by moving that phosphate. So we're going to use what's known as a mutase. So we use phosphoglyceromutase to move that phosphate group. Still on the same molecule, still a glycerate molecule, but now it is 2-phosphoglycerate. Some of you may be scratching your heads and asking, well, why did we even bother doing this? I can tell you, one of the main reasons why we bothered doing this is that if we did not move that phosphate, the enolase, which is going to remove some of the water and get us closer to pyruvate, would not recognize it. You've all done the enzyme lab now, and you know how specificity works. This enolase, which is involved in converting this 2-phosphoglycerate into phosphoenol pyruvate by removing water, would not function if we didn't have that phosphate on the 2 carbon instead of the 3. Enolase is going to result in the removal of water from this 2-phosphoglycerate, giving us phosphoenol pyruvate. Okay, you can see the water molecule leaving right there. Okay, so we have phosphoenol pyruvate, and we now are going to use pyruvate kinase to remove the final phosphate group and leave us with pyruvate. Hooray! Drinks on me after class. Just kidding. All right. Now, this is only the first of 3 different processes that we have to talk about today. So, how's everybody feeling at the end of glycolysis? Beautiful, yeah. Now, guys, in all seriousness, though, I kid because I can already tell that this has taken its toll. But in all seriousness, let's sit down and think about this, okay? Because half of test taking is actually studying, the other half is figuring out the kinds of questions your instructor could potentially ask, okay? Which is why if you only study a little bit, you typically wind up with 50. Now, but seriously, let's think about it, okay? The big things that you need to look at in this part of the process are the NADH being produced and how much of it we are producing. Okay? The names of the enzymes that correspond to each of the intermediates. The amount of ATP we're producing. The fact that this is energy payoff. You'd be surprised how many people cannot identify for me energy payoff or energy investment. Now, the other thing I need to ask you, just kind of a review. What kind of phosphorylation is occurring here? Is it oxidative or substrate level?
Substrate.
Substrate level. Remember, glycolysis and the citric acid cycle both utilize substrate level phosphorylation as a means of phosphorylating ADP into ATP.
Out of curiosity, what is that substrate?
What is the substrate?
Yeah.
You're looking at it. Phosphoenol pyruvate, in this particular instance, would be one of the substrates. That's the substrate that the enzyme recognizes. And ADP would be the other substrate.
So this is just sort of floating around out there in the fluid.
The enzyme?
Yeah. Well, the substrate actually. That would be located.
Phosphoenol pyruvate?
Yeah.
In this particular instance? Yeah. If you remember also, glycolysis is the only stage that takes place in the cytoplasm. Once we hit the citric acid cycle, we're now inside the mitochondria. But yes, to answer your question, Richard, there are enzymes floating around when this product is produced. They're going to bind to it. They're going to facilitate, speed up this reaction so that we produce pyruvate and ATP. There are also enzymes that can be inside the mitochondria during the citric acid cycle. Yes, Kate? This particular process, well, actually, y'all saw, it doesn't really take that long at all. Y'all watched fermentation occur over the time span of 40 minutes, and you notice that you produce a large amount of carbon dioxide in that 40-minute time span. So typically, this takes minutes to occur like that. Now, we did make ATP. Woohoo! Remember, that's kind of the overall thing we're talking about with respiration, is the production of energy. But we only technically made 2 net ATPs. But here's the important thing. Through this process, we also made NADH and those protons. You are going to hear me talk again and again about the NADH and those protons, because they're going to play a major role in oxidative phosphorylation. I cannot stress to you enough how important these 2 things are during both this process and, as we'll see, in the citric acid cycle. And you will see very, very, very soon why they're so important. But I don't want to spoil the surprise.
We made NADH from the NAD. Where did we get the NAD?
The NAD is a coenzyme, which means it is an organic compound that's probably a vitamin or something similar to that. Okay? We can generate it, in theory, inside of our cells, or if it's something we can't generate naturally, then we'll need to actually synthesize it or consume something that allows us to produce it. It's a great question, though. I don't actually know the origin of that molecule.
Okay.
But I know that we do produce it. Okay. So, questions, concerns about glycolysis at this point? Yes?
Is it the same to know that NADH produced for how much? And what was the next thing you said?
You're going to want to know the amount of NADH produced. You're going to want to know how to name the enzymes or recognize and identify what enzymes bind to which of these intermediates or substrates. You're going to need to know the total or gross amount of ATP produced during this process. You're going to need to know the net amount of ATP produced throughout the course of glycolysis. You're also going to need to be able to tell me, based upon pictures, are you looking at energy investment or energy payoff? What's the easiest way to do that?